Hello, everyone, and welcome to this episode of Foundry Fireside. My name is Jeremy, and I'm an intern working on the Foundry communications team. Today, I'll be talking with Aeron Hammack, a staff scientist who has been working on genomic sequencing pipelines and assay development. So, Aeron, could you please introduce yourself and tell us about your project?
My name is Aeron Hammack. I'm a staff scientist at the Molecular Foundry, and I work on biological nanosensing, so leveraging nanostructured inorganic semiconductor devices to measure the properties of individual molecules or single biological entities, um, using all of the incredible scientific capabilities we have here at the Foundry.
Okay. Um, so why did you choose this research?
So for the past 10 years, I've been working in biopharma on bacteriophage-based pharmaceutical development to combat antibiotic resistance in bacterial infections. And along the way, we did millions and millions of compatibility assay between bacteriophage, which are viruses that only attack bacteria, to assess whether they would be useful as potential antibiotic compounds in formulation. However, the current standards for the assay development that you use in biopharma development rely on lots of plasticware, plastic labware, and fairly large volumes of liquid, much larger than the amount required to house a bacterium happily. So along the way, became passionate about methods for assay miniaturization and trying to get down to not only looking at millions of bacteria together, but individual bacteria or the individual molecular components of the bacteria and the viruses that attack them. So that was the motivation.
All right. Um, could you explain what an assay is really quick?
So assay is a fancy word for a test that gives you back some extra information about the composition of an unknown liquid or an unknown sample from your environment. So if you've gone to any lab, like a lab core testing services facility, or your doctor runs a diagnostic on you, those are types of biological assays, and they'll test for different compounds that might be in your blood or different compounds that might be in the water that we drink. So those are incredibly useful tools for figuring out what's in our surroundings or what's inside of our bodies. It's a fancy word for test.
Yeah, yeah. How do you think this research is going to affect or is currently affecting people kind of outside the science community?
So the historical research that I did in the in industry is actually in clinical trials right now, which is affecting people by treating urinary tract infections. And then the work that we're doing at the Foundry will be directly relevant in the sense that we're working on improved tests or assays for the bacteria versus bacteriophage compatibility, but using much smaller miniaturized liquid handling techniques. And the big difference is for the pharmaceutical development programs, we literally used millions of plastic containers, tiny plastic containers, to do all of the tests. But if the as the research becomes successful for what are called microfluidic assays or nanofluidic assays, you don't have to use all of the plasticware. You can use multiple use semiconductor devices or chips to do the assays at much smaller volumes with less waste, which is great for increased sustainability when doing pharmaceutical research.
So you're looking at how we can like reduce the amount of plasticware by reducing the size of the bacteria or viruses versus like whatever you're
Yeah,
treating them with.
yeah, reducing the amount of liquid that you need to test.
Hmm.
So if you need to test a whole bucket of liquid to get your answer, there's a lot more waste associated, both in the reagents that go into it, the liquids that you have to prepare, as well as in pharmaceutical development. You normally use single-use sterilized plastic components. So if you're throwing away one full bucket for every test that you run, that's a lot of plastic waste. Sure, there's recycling, but recycling takes energy. By trying to miniaturize the amount of liquid required to perform the evaluation, then you can minimize the amount of waste that's consumed when you're performing your biological tests. And then the other thing is, in addition to just the sustainability component, there are fundamental scientific questions that you can only answer if you're looking at one virus at a time or one bacteria at a time, instead of looking at millions and millions of viruses all together or millions and millions of bacteria all together. So it's the question, we all just lived through the coronavirus pandemic, and everybody's familiar with it's the variants that lead the wave of infectivity. Well, each one of those variants, it starts out as a single virion, not a collection of viruses. So if you want to have really early phase detection, you want to be able to detect at the single particle level what's the pattern of emergence of these genetic traits arising from one virus at a time as opposed to a collection of viruses at a time. It's a signal to noise question of how many observations does it take to know what's happening in your surrounding environment. And by testing much smaller volumes of liquid, then you know that you're actually testing a few viruses instead of millions of viruses, or a few bacteria instead of millions of bacteria, which means you can find the individual traits as opposed to the community traits.
So in doing these tests, what equipment are you using and kind of how does it work?
So for the tests themselves, there's delivering the liquid. So it's pipettes, which are fancy syringes that allow you to dispense very precise amounts of liquid. Capillary tubes, where you're similar to the plumbing in your house, just on a much smaller scale, with tiny spigots and faucets to deliver them to the actual semiconductor chips themselves. And the semiconductor chips themselves, that processing looks more like what you would use to make a computer chip that's inside of your graphics card or inside of your CPU sitting on your desk or in your laptop. So you're dealing with all of these normal plumbing and pipetting and syringing, kind of large scale liquid handling, and then you're piping them in to semiconductor chips that have what are called lithographically patterned microchannels or nanochannels in the surface that then take the liquid from the larger scale plumbing and then route it around inside on the chip using electric fields or pressure to miniaturize the assay to where you're looking at tiny microscopic or nanoscopic droplets of water or solvent that the biological material is dispersed in. And so those would be the main kind of larger scale, what we're doing directly in the foundry to create new things. And then the other sorts of equipment that we use would be optical microscopes or electron microscopes, just to look at the samples and characterize them, or genetic sequencers to figure out what the genetic components of the viruses, the bacteria that we're testing are.
So when you're looking at droplets of water this small, are you using microscopes or what else are you using to kind of observe what's happening on these chips?
You would be using microscopes as well as the electrodes that are embedded into the chips to read out what the change in the electrical conductivity of a material is when there is or is not a biological reagent present. There are things called affinity assays where the readout from a CMOS device will change depending on whether or not a biological molecule is bound or unbound from an electrode inside of your circuit. And there are ion currents where as a biomolecule flows through a tiny channel, sometimes it'll stop, plug up the channel. And normally if you're using electric currents to push it through the channel, what happens is the biomolecule will plug up the channel and the ions won't flow. So you can see, hey, this channel is plugged by this biomolecule. And you read the difference in the ion current to make a best guess of what was plugging up the hole. And then similarly, like you mentioned, optical microscopes, where you can tag your molecules with fluorescent dyes or fluorescent proteins to keep track of where they're flowing inside of your circuits. Or you can look for what are called vibrational spectroscopic signatures, which is you're reading out optically without labeling the molecule what the internal vibration states of the molecule are through a techniek called Raman spectroscopy. So there's a number of different ways that you do the readout, and the devices that we're working on enhance the sensitivity with which we can tell which molecule is most likely to have been in an active region next to an electrode or next to what's called a plasmonic antenna, where plasmonic antennas are tiny optically active antennas that can deliver visible light information into your microscope objective so that you can collect the spectral signal of your biological compound. And then the idea is you collect a library of all of the different types of spectra that you get from all sorts of different biomolecules that you might be interested in, or biological entities that you're interested in, so that you can say, oh, okay, I saw one of those before, it had this fingerprint. I saw one of these before, it had this fingerprint. And now I can mix and match and tell what's actually inside of my tiny droplet of liquid that I've put onto my functionalized chip.
Okay, very interesting. Um, what makes you excited about this topic?
This might be a bit of a nerdy excitement, but I started out my research as a low temperature physicist, and when you're doing physics, there's a discipline of what's called statistical mechanics, where you try and keep track of all of the emergent properties of a large collection of objects, so gas molecules in a room or atoms inside of a crystal. And there are these fundamental challenges in differentiating the behavior of the individual gas molecule that's in the room versus the thermodynamic uh, bulk properties of the molecules in the room. So being able to keep track of individual particles like individual photons, we have individual photon counters, we have individual molecule counters. So just bringing that level of precision in characterization of the individual objects to biological assay development is very exciting to me, because it's just a whole different level of characterization when you're looking at things one at a time and then describing the entire population by picking out each one and characterizing each one individually to take an image of the whole population, as opposed to just figuring out, well, what's the temperature in the room without knowing where each of the atomic or molecular elements that contributed to that temperature, what they were doing individually. So just having that increased precision should open up whole new avenues of just biological exploration and understanding, to know what's actually happening inside of a cell that's living, or what happens inside of cells that die, what makes a virus, but just that one virus successful, not the whole herd of viruses. What makes an individual virus successful? That to me is exciting, because it's just getting into a level of detail that has only recently been available in the field of biology recently on a scientific scale. So within the last 20 years, but still relatively new.
So kind of to lead off of that one, what new questions do you think your research is going to open up?
Um, That's the tough one. But what I really hope is that as we build these nanosensing modalities, we can get to a level of characterization, sensitivity, and precision that we can start to build things like protein sequencers. So we have a lot of DNA sequencers, and we've recently developed direct RNA sequencing technologies where you can measure the sequences inside of individual RNA molecules. But to date, most of the protein sequencing technologies, they're using mass spectrometry and tying them to associated messenger RNA structure to have a best guess of what the protein sequence is, through a combination of computational analysis and looking at the other molecules, but not the direct sequencing of the protein itself. And some of the nano poor people have started to do direct protein sequencing, which shows that it's feasible. And then as we improve our capability to do these biological nanosensing assays, just being able to look at the individual protein components inside of all of the cells and sequence them individually, so you can kind of take the puzzle apart and look at all the pieces, the puzzle being the cell, and then figure out how to fold them all back together and stick them back together. That's kind of one of the grand scientific challenges, is what makes a cell tick. And then by improving our capability to sense the puzzle pieces, it's easier for us to understand how it all fits together. That to me is the most exciting thing, is just what makes a self-replicating organic system continue to self-replicate in its environment, and just getting down into the weeds of characterizing each of the pieces. And what is life? It's a huge question, and an exciting one. It's kind of like maybe we'll figure that out someday. Not
Mm.
tomorrow.
Yeah. Um, is there any new research that's happening right now, kind of outside of your field that you're really excited about? Or maybe it is affecting your field, but is there anything that new that excites you?
Yeah, I'm really excited about all the quantum materials characterization that we do at the Foundry, because it turns out that the types of devices you build to make qubits or quantum information processing systems, they look a lot like the types of devices I want to build for biosensing. So being able to participate in a community that has strong quantum information science and quantum materials programs in order to leverage all of the uh, you know experimental measurement techniques that we have is really exciting. And it's also exciting because that's kind of where I began my journey as a physicist, was working on um, degenerate gases of bosons as potential substrates for quantum information processing in the future. And it turns out, when I started like 25 years ago, the future was the future, and the future is now, where there are actually companies working on building quantum information processor systems, which has just been it's fun in your career to see something that you worked on a long time ago start to actually hit commercialization as devices that people may be using to do actual computational problems. So that's really exciting.
All right. Is there anything else that you'd like to add?
Um, Nothing that I can think of other than the Foundry is one of the things another thing I'm really excited about at the Foundry is the degree to which it serves as kind of an extra educational institution. Case in point to this interview, where we have a lot of awesome internship opportunities or postdoctoral opportunities for people to come here and work and not just do world-class science, but learn how science itself works. The Department of Energy is just a great place to learn how the U.S. has done science and big collaborative science. Like it's really awesome to work in a highly collaborative scientific environment where when you walk down the hall, you can find any number of world experts on a wide variety of different disciplines, from chemistry to biology to quantum mechanics to material science to spectroscopy, all sorts of different just fun avenues of research.
Okay. That's all the questions I have for you today. Again, thank you so much, Aaron, for agreeing to meet with me. It was a pleasure to learn more about your research. And for everybody who's tuning in, thank you for watching, and look out for more episodes of Foundry Fireside on our website. Thank you.
Thanks. Thank you for the interview. Appreciate it.